Hi again, biologists. At the end of part 2, we were discussing the scientific method and the different parts of science. So I wanted to show you a little bit more about analyzing data. So generally, when scientists collect data, they graph it, they analyze the data with statistics, they look at what's the chances of happening randomly versus that there's actually something going on here. So this is an example of a graph. And what you do is you graph the independent and dependent variables on the x and y axis of a graph. So what does independent mean? Temperature is an independent variable. That means it doesn't depend on anything else. So if you were looking at how many flatworms there were at different temperatures, you would look at 7 degrees Celsius, 8, 9, 10. Those are all independent. The dependent variable depends on the independent variable. So the number of flatworms is based on the temperature. So they've plotted that graphically. Each of these points is a data point. So you see the temperature was about 6.7, and the number of flatworms there is about 72. So we have this grouping of data graphed. Then what you draw is a regression line. So you can plot this in a program. That shows you a general representation trend. Sometimes you'll even see it just called a trend line. So what we have is we have a negative trend, saying that as the temperature increases, the number of flatworms decreases. So that's called inversely proportional. As one increases, the other does. If, on the other hand, we saw a line that as temperature increased, the number of flatworms increased, that would be a positive correlated value. As one increases, so does the other. Inverse, as one increases, the other decreases. And we use what's called an R-squared value to see how well our data fit the line that we've made. So you can see our R value is 0.96, R-squared value. It's very close to 1. 1 would mean there's a perfect correlation. Every single one of those points would be exactly on that line. So 1 is the highest value, 0 is the lowest. So if there was 0 correlation, those points would be all over the board. That line wouldn't really make sense. So we have a high R value. That means those two numbers, those two factors, temperature, is very well correlated with the number of flatworms present. Okay, and people do, as we come out with new science, one of the great things is that we are willing to change our mind based on new evidence, but that can also be frustrating. Oh, they said this and now they say that. But there is a rigorous process that any scientific conclusions have to go through before they get published. So there are a lot of scientific journals out there, and there are some that are very hard to get your research published in and that we take very seriously. Those have what's called a peer review process. In peer review, you'll have other researchers in the field read through the paper and say, is this good science? Is it not good science? What should they have done differently? What do they need to say? Are they making conclusions that they shouldn't be based on the evidence they collected? We also try to do controls. So if you're looking at how that temperature changes flatworm numbers, you'd also want to do flatworms just at like their normal temperature to make sure that that population of flatworms is healthy and that it wasn't just that they were dying because they weren't healthy. Okay, and I talked a little bit about a scientific theory. Scientific theories are these overarching statements that explain the facts that we're finding. They've been tested over and over and never proven wrong. So example, cell theory, we all come from cells. Atomic theory, that there are small atoms with protons, electrons, and a nucleus. The heliocentric theory, that the Earth revolves around the sun. And the evolutionary, that living things evolve and come from common ancestry. So how is a theory different from a law? So a law describes patterns that are seen in nature. What does that mean? That means that you're going to have a mathematical equation that you can use. So like force equals mass times acceleration. the coefficient of friction affects how things move. It's going to be math. Okay, that is the end of Chapter 1 PowerPoint lecture. And now we'd like to show you a little bit more on how to do the Chapter 1 homework assignment. Okay, I'm on our Blackboard site, and here's the MyLabs Plus. And what I didn't know, this is new with the new version, is you're going to want to open this in a link, open link in a new tab, just because otherwise you've lost your Blackboard. So I like to open it in a new tab. And then here's our course. So you can see over here, we've got the introduction, chapters 1 and 2. So I'll go into the student view. Okay, so here are the different questions that you have to answer. A lot of them are going to be like animations, listening to a little bit, couple of general questions. So what you need to do is you need to go into each and complete them. And realize that each time you submit an answer, if you get it wrong, it's going to take a percentage off from your grade. So try to go through these slowly. So click to start the animation, look through it. You can work through there, look at all the different parts that make up the characteristics of life, and then answer the questions. Double check them in your book, look at the part of the PowerPoint again to make sure that you're getting them right, because you will lose a proportion of your grade for each you get wrong. And then, I'm moving this over, you can go on to the next question and work on that. So up at the top you'll see the difficulty level, 1 means the easiest, and the time on average it takes students to do. So when I'm working on these assignments, I can see the generally average time it should take a student to complete, and I try not to make it more than an hour, because I know that sometimes students take longer than what it says. So complete the activity, answer the questions. You won't get a grade on it until you've finished all the parts. Okay? And then when you're done, if you would like to look at your grade, you'll have the grade over here in scores. I don't have any grades, so that's what it's going to tell me. But there you can see what you've worked on in your total score. I think it takes longer for the MyLabs Plus to update into Blackboard, so you might not see those items go over for up to probably a week. So even if they're not in your Blackboard grade, you'll be able to find your scores here and you can calculate those. If you have any problems calculating, you can ask me. Okay? And then once you finish the homework assignments, I'll post the quiz over chapters 1 and 2 and you can work on that. And you have unlimited attempts on that to try to get as good a score as you want. I'll probably have about 60 questions and it'll ask you 20 of them each time. Have a great day!